junction. So this is the synapse between our postganglionic neuron and the effector tissue in question. So it's also a synapse. Excuse me. So we're going to see a lot of the same events that we talked about at a traditional synapse occurring here as well. But this is going to be a little bit different from a traditional neuron-to-neuron synapse, because here we're synapsing from a postganglionic neuron, which is that second neuron in our series of 2 neurons, and the effector tissue or effector organ that it is innervating. So this can be cardiac muscle, this can be smooth muscle, this can be adipose tissue, or glandular tissue. And so 2 important differences that we see between a neuron to neuron and the neuroeffector junction is that we've got these swellings along the axon terminal. So we don't have a discrete axon terminal the way we've been drawing it. What we see here are varicosities. So varicosities are these swellings that are located at the end of this postganglionic neuron. And this facilitates the release of neurotransmitter to innervate this effector organ. The second difference that we see here at the neuroeffector junction is that the distance between the synaptic cleft, where we have this varicosity, and the effector organ is much greater than it is for a traditional neuron-to-neuron synapse. So I'll say that in another way. When 2 neurons are synapsing to each other, we talked about that synaptic cleft, that distance is much shorter or much smaller than the synapse between a postganglionic neuron, the varicosity, and the effector tissue. So those are 2 important differences between a neuron-to-neuron synapse and the neuroeffector junction. Okay, right. So let's look here at what happens between the varicosity. So I'm going to zoom in here. So this is showing that varicosity. And before we look at the events, I want to point out that we've got voltage-gated sodium channels up to the varicosity. So this is where our action potential is propagating. And then at the varicosity itself, we have the voltage-gated calcium channels, just like what we saw at the axon terminal when we looked at the neuron-to-neuron synapse. Okay, we've also got synaptic vesicles within the varicosity, and this is where our unique neurotransmitter is being stored. So that can be acetylcholine if we're speaking about a postganglionic neuron in the parasympathetic system, or it can be norepinephrine if we're speaking about a postganglionic neuron in the sympathetic nervous system. Okay, so make sure you're keeping those 2 divisions distinct or separate. All right, so let's kind of draw this out here a little bit. So we're looking at the neuroeffector junction. And in brackets here, I'm going to put that this is between a postganglionic neuron and the effector tissue. Now again, bear in mind, this can be either the sympathetic or the parasympathetic division. Okay. So I'm going to draw what our neuron looks like. So we've got this postganglionic neuron, and instead of ending at a discrete axon terminal, it's going to end in these varicosities that I talked about. So these are the ending of the axon terminals, and then we've got these swellings along the endings that are called varicosities. Okay. And if we zoom in really closely to look at what's happening between the varicosity and the tissue it's innervating, so let's draw some tissue here. Right, so this can be a piece of smooth muscle. This can be an adipose tissue. This can be glandular tissue. For our purposes, let's just say that this is smooth muscle. Okay, and so we're zooming in between the varicosity and the smooth muscle itself to see exactly what takes place at this neuroeffector junction. Oops. Okay, so this is the varicosity here. And then this is our smooth muscle on the other side. Okay, so we know the first part of this is that an action potential is traveling down this postganglionic neuron. So let's label this out as a postganglionic neuron here. And we know that an action potential is traveling down the length of this neuron in this direction. Right, this is our action potential. And that's step one. Oops. Right, so the action potential travels down neuron. Okay, the second step is that when it gets to the varicosity, it's going to trigger the opening of voltage-gated calcium channels. So this is something we know from prior knowledge. Along our varicosity, we've got these voltage-gated calcium channels that are facilitating the movement of calcium. And so the second step is that when the action potential gets to the varicosity, we trigger the opening of this channel and calcium rushes in. Okay, so that's step two here. So the action potential triggers opening of calcium channels. And this causes calcium to rush into the varicosity. Okay. So far, this should look very familiar to what we've spoken about before, right? We talked about the events at the synapse, and so all of this should really be a review because the events are very similar to what happens at the synapse. We're just adding a little bit more detail here. Now, within this varicosity, we know we've got these synaptic vesicles that are storing our transmitter, our neurotransmitter. And this neurotransmitter can be different depending on where we are. So if this is in the sympathetic system, this neurotransmitter is norepinephrine. And if we're in the parasympathetic, that's going to be acetylcholine. So just bear that difference in mind. We're going to draw it in terms of just the general events, but understand that this can be a different neurotransmitter depending on where this neuron is. And so the third part of this is that the calcium rushes in and it triggers the release of these synaptic vesicles via exocytosis. And so these neurotransmitters are dumped into the cleft, okay? And so that's step 3 here. All right, so step 3 is that we've got our neurotransmitter being dumped into the cleft. Step 4, it happens on the membrane of the smooth muscle. And this is where we've got our receptor, right? Remember, we have a receptor that is unique to this neurotransmitter. So if it's acetylcholine, it's going to be a muscarinic receptor, so not a nicotinic receptor, because we know that nicotinic receptors are not found on effector tissues. Nicotinic receptors are only found on the soma of the second neuron, so on the soma of the postganglionic neuron. So here must either be a muscarinic receptor if this is in the parasympathetic system, or an adrenergic receptor if this is in the sympathetic system. And so a muscarinic receptor will bind acetylcholine, and an adrenergic receptor will bind norepinephrine, okay? And so our neurotransmitter binds to this receptor, and that's the 4th part, or the 4th step here in this sequence of events. So we got step 4, the neurotransmitter binds to its receptor. Alrighty. And then the fifth step here is basically cellular response. Okay, and we talked about what would happen if this was a adrenergic receptor. So if this was an alpha-1, alpha-2, beta-1, beta-2, or beta-3, we know what the cellular response would be. We talked about the specific signal transduction pathway for each of those receptors. And so we know how the cellular response would come about. So we're not going to go into more detail with that here. But understand when the neurotransmitter binds to this receptor, it's coupled to that G protein. And this is where that signal transduction cascade will come about to bring about our cellular response. And then step 6, again, we should be familiar with. This is where we have the degradation of excess neurotransmitter. That is brought about by our degrading enzyme, which is acetylcholinesterase. So we've got this Pac-Man type enzyme here, and that is called acetylcholinesterase. And that is going to degrade our excess neurotransmitter and help to clear the junction. There should be an E. Help to clear the junction of any excess neurotransmitter. And then that can be reuptaken back into the varicosity and recycled to make more neurotransmitter. Okay, so this sequence of events we kind of already touched on. So the general flow should be very familiar, but we're just adding in some detail as it relates to the neuroaffective junction to the autonomic nervous system specifically. Alrighty. Okay, I hope this is not too confusing. So I'm going to go over this one more time on our slides, and then let's save this. Okay. And then we'll see if any questions come up after that. Okay. All right. Okay. So here is that kind of summary of what we talked about just now. We've got our action potential, triggering the release of calcium. Calcium rushes into the cell. This triggers that exocytosis of our neurotransmitter. And now this is very nicely showing what both of the postganglionic neurons would look like. So if we're in the parasympathetic system, this is what it would look like. Our neurotransmitter is acetylcholine. Our receptor is a muscarinic cholinergic receptor. If we're in the sympathetic side, this is what our receptor, our junction would look like. Our neurotransmitter is norepinephrine, and our receptor is one of the many adrenergic receptors, so alphas or betas, okay? And then this brings about some type of cellular response on the tissue in question, depending on exactly where that tissue is. Now, I'll point out one other important difference here. So the way that we clear the excess neurotransmitter from the neuroeffector junction, from the cleft here, is by breaking it down with this enzyme acetylcholinesterase. But understand that acetylcholinesterase can only break down acetylcholine. So in the instance of norepinephrine, we do not have this acetylcholinesterase enzyme. We simply have norepinephrine in its entire state being reuptaken back into the varicosity and being recycled to make more norepinephrine synaptic vesicles. So the acetylcholinesterase enzyme that breaks down acetylcholine to acetate and choline is not seen on the sympathetic side because it does not break down norepinephrine. Norepinephrine, as it is, can be reuptaken into the varicosity. It can be repackaged into a new synaptic vesicle, and that can go on and on, okay? All right, so anytime you see acetylcholinesterase, automatically you know you're looking at the neuroeffector junction on the parasympathetic side where we have acetylcholine as our neurotransmitter. Okay.